Thank you for the introduction. I'm Yicong Fu from Cornell University. I work for Sunny in his lab. I'm a fourth-year PhD student studying fluid structure interactions. So today I want to talk about fish and how I got inspired from fish to study these interactions between fluid and solid structures, especially with perforated flexible panels. Yeah, over the years, people have been inspired by fish a lot, especially with their locomotion. So what people did is, from the observations on the actual organism, engineers or experimentalists simplified them into hydrofoils and put them into water tunnels for experiments. And then at the same time, we have mathematicians who are conducting simulations based on computational fluid dynamics models. And then we also have theorists who work on mathematical models, such as Lighthill's model, that calculates how much force that the fish can produce based on energy balance. There are also Euler-Bernoulli beam theory to calculate how much deformation that there can be on the solid structure. And then there's also added mass term for sort of a scaling to measure how much force that the fish can produce. And with all of these knowledge, I was very privileged to be able to work on such robotic systems that mimic the fish locomotion strategy and turn them into something that people can use. However, if you look at the history, people have been making some pretty strong assumptions. And these assumptions, to me, are not necessarily completely realistic. So people either work in high or low Reynolds number regime. People usually make a lot of 2D or quasi-steady assumptions. And then, especially with the CFD, a lot of the kinematics are prescribed, which means there's no true coupling between the solid and the fluid. All right. And then, of course, the geometry usually is very simple. People usually use just a rectangular piece to simulate what the fish is. However, I want to propose a question. So if we lift these assumptions, what other physiological aspects can we still study from these fishes? In other words, if we have three-dimensional flow, if we work in intermediate Reynolds number, where inertia and viscosity are both important, if we have active motion from the muscle on some part of the structure, and then the rest is just passive deformation, what's going to happen? And then what if we have complex geometries that are more crazier than just a rectangle, right? So I looked at fish and I thought, fish gills are really interesting. Right. So these are respiratory organs on the fish. And if you look at the microscopic structures, they are perforated. They have these primary lamella and the secondary lamella that intertwine into this mesh-like structure for cross-flow ventilation. Okay. And then biologists have also found that they have muscles. So the fish are able to control the actuation, its motion, for its purpose. And then more interestingly, these structures, especially the primary lamella, has flexibility. And the flexibility depends on the inflow mechanism. So in general, fish has two inflow mechanisms. The first one on the top is called orbicular pumping. So they use this periodic motion of the gill covers to create oscillatory pressure differences to draw water in from their mouth and then expel them from the gill slits. And then the second type on the bottom, the shark, is called ram ventilation. So these fishes have to swim continuously forward with their mouth open, and the water just rushes in to the gill cavity. Okay? And then some other biologists have found that these gill filaments, the red part, are actually calcification. So they have some level of rigidity or flexibility depending on which mechanism they choose to use. And there's a trend that the first mechanism of inflow has less calcification. So they're more flexible than the second type. And people have sort of believed that this is due to unnecessary deformation of the gill filaments. So the gill filaments, we can think of it as this way. So these are different sets of gills. And then for the cross-flow ventilation, water comes in from here and then crosses over the gill filaments for respiration and then exit out, right? And then nominally, the endpoint of the gill filaments are sort of touching together. So all of the flow are forced to go either on left or the right side for respiration. But for the ram ventilation, there's so much force pushing on these filaments that the ends are actually bent and separated. Right? So they think that there is a certain amount of shunting flow going in the middle that's not participating in respiration. So that's not good for the fish, which is why they need calcification to reinforce it. However, me as a mechanical engineer asked a question, which is, it's possible that it's not completely true in biology. To me, it's not very difficult for animals to make something that's completely rigid, like bones. Fish have bones too. So why in evolution did they not produce something that's completely rigid so that this endpoint is always fused, right? Why do they still retain the flexibility? So that's the biological question I want to propose. And then we can also translate that into fluids problem, which is how do perforation, actuation, and flexibility affect the hydrodynamics of these hydrofoils, and then also the associated transport phenomena.
Sorry, what is a hydrofoil?
Hydrofoil, like, so basically that piece.
Okay, good.
Yeah, it's called hydrofoil. Yeah. Yeah, so I conducted experiments. I made these mesh-like structures out of two different materials. One is silicone, which is the flexible case, and then the other is 3D printed PLA, which is the rigid case. And I put that onto a motor that can be actuated and then in a flow tank so that we can study how it moves the water around it in these PIV experiments, which is particle image velocimetry. So first, let's take a look at what the perforation does. So on the left side is the non-perforated panel, which is like a very classical problem that people have already studied. So if you look at the flow structures, it produces this large vortices in the wake, and there's a single large vortex over here. So in our field, we call it 2S wake. And then the accumulation of these vortex structures behind it is actually a combination of the trailing edge motion, creating this vortex, as well as the leading edge vortex that's created in front of it and then rolls all the way down to the trailing edge. So they merge together. However, if we look at the perforated case, sorry, playing the video. Sorry. Yeah, we see a completely different vortex structure in the wake, even though the prescribed motion is completely the same. So we have two P wakes. So every stroke produces a pair of vortex that's counter-rotating. And then there's a lot of flow that's penetrating the panel because of the perforations. So there is more transverse momentum of the fluid than that case. And then we can also take a look at how actuation changes the flow field. So on the left side is the flexible perforated case, sort of just situated at this angle without any motion. So it acts just like a rigid panel. It's not moving. There's no oscillatory behavior due to the vortex. Well, in fact, there is no vortex. You can see that all of these flow just go through the holes and then into this laminar meandering wake, right? But however, on the right side is the actuated case. You already saw that. There's a lot of vortex shedding, interactions between the vortices, creating this flow field. So as I said, the fish gill is for material transport, right? These transport phenomena. So let's think about these structures are releasing carbon dioxide, for example. So after I get the velocity field from these experiments, I can put that into a simulation to calculate how the concentration of material would look like in a convection diffusion equation. So these are the results. On the left side, well, both of them actually resembles what I saw from the experiments, which is great. This is the simulated case. This is sort of a mass transport, right? So we can see that on the left side, if we don't have motion, the mixing or the dispersion is much worse than the right side where we have motion. And then we can also look at how different types of actuation, or especially the frequency of pitching, changes the behavior. So let's take a look at the rigid case first. When we pitch it at a low frequency, we have this 2P wake that we just saw. Yeah. And then when we have a higher frequency, we still have the same wake structure, but you see that the dispersion is actually narrower. So even though we input more energy into the motion, the effect that we get is worse. However, if we have, flexible cases, we are able to maintain this good mixing or transport result by changing the wake structures. So it transitions from that centered 2p wake into this bifurcating, diverging 2p wake. So going back to the questions that I had, from the biology side, why do they still need flexibility? Maybe it's because they want to induce these vortex transitions to maintain a good material exchange across different parameters. But of course, there are a lot more to be done to really study this. And then on the fluid side, how do we model such complicated systems when we have these shear layers sort of interacting with each other and combining into bigger wakes? Is this just a superposition of different flow path cylinder models, or is it more complicated than that? It's something that people can work on. And even more, if we look at other organisms in biology, the fish tail, the antenna of the moth, the root system of vegetables, and the fungus, porous fungus, creating condensation, these are all porous structures that interact with fluids. So they're really interesting to study. And then last but not least, I just want to say I love fish so much. So I put them in my career, in my life, and in my stomach. So yeah, well, thank you for listening. I'm happy to take any questions.
I guess while you can answer some questions, Rain, why don't you get set up and then we just like question transition time.
Yeah.
Yes, yeah, please.
The basal assumption in the work is that there's this one driving evolutionary force, which is about fluid flow over the gills. It seems reasonable to me, but are there other reasons that gills might be shaped? Are there other?
Yeah, of course. Yeah, the gill is actually a multifunctional organ. So the gill structure, well, you can split it into three parts: the gill rakers, gill arch, and gill filaments. The filaments is for respiration. But the gill rakers, for example, is for particle capture for filter feeding fish, also protection of the cavity within. So there might be some interactions between how that purpose and the respiration purpose drive evolution together to what they look like right now. And I agree, it's very complicated. Yeah.
So in your simulation, what was
Yes.
What was your Reynolds number?
The Reynolds number was 500. Yeah.
Can you give us like an intuition of like
Is that big or small?
what do we mean by 500? Like what?
Oh yeah. So 500 Reynolds number is like sort of in this transition zone where you start to have vortex shedding phenomenon. But in my problem, I have these small gaps between the little posts. And over there, the Reynolds number will be much lower. So it's a different scale problem. So over there, it will be more viscous dominant, but overall, it's more inertia dominant, which is why it's so complicated and interesting to study, I think.
When you were driving,
Yeah.
like the perforated sheet versus the full sheets, like
Yeah.
what did you keep constant? The force that you executed on the sheet, or like if you wiggle a less material containing sheet?
Yeah, great question. So the way I actuated it is keep the angle range, but change the frequency. Yeah, but to answer your second question, if it's more flexible, it actually actuates a different mode of the flexible structure. It's like a standing wave, for example. So around the panel, I see more of a stagnant water zone, and then the external flow just go around it. And then the whole flow field becomes like a bluff body, and then behind it is just a slowly recirculating vortex. So I didn't put that into my presentation, but from what I can tell, it's less mixing or worse transport than these vortex shedding cases.
Very interesting.
Yeah.
Thank you so much.
Thank you so much again.
All right.